This protocol enables researchers to monitor survival on a single-cell basis and identify variables that significantly predict cell death or survival.
Conventional cytotoxicity assays assess populations of cells and lack the ability to discriminate individual cells. Longitudinal microscopy allows the assignment of time of death for each cell in a population.
This technique is well suited for evaluating new therapies for neurodegenerative diseases and other conditions.
This method has proven invaluable for exploring disease mechanisms and neurodegenerative conditions, and could be applied with equal efficacy towards other disorders in which cell death is of interest.
Researchers may struggle with achieving adequate transfection efficiencies without undue toxicity. Practice with the multi-channel pipette and familiarity with the protocol should diminish these difficulties over time.
Microscopy is an inherently visual methodology. Therefore, the technique may be more easily understood by watching in addition to reading.
To begin, combine the appropriate amount of reduced serum media and transfection reagent in one tube and reduced serum media and DNA in a separate tube. Incubate them at room temperature for 5 minutes. Then, combine the contents of both tubes and incubate at room temperature for 20 minutes. Use a multi-channel pipette and sterile plastic troughs to wash the cortical neurons 2 times with 100 microliters of neurobasal media per well. And store the conditioned media and other remaining medias at 37 degrees Celsius. Replace the NBM with 100 microliters of previously prepared NBKY solution per well. After 20 minutes, pipette 50 microliters of the transfection reagent and DNA mixture.